If you're feeling a little bit confused, stressed out, or maybe just curious about what makes an operon positive inducible or negative repressible, hang out for a few minutes. Hi biology friends, it's Sarah, your other biology professor, and let's talk about some categories of gene regulation. We're talking about regulating gene expression, the process of going from gene, DNA, to a functional product, maybe an RNA, maybe a protein. Now, cells don't need all gene products all the time. So they regulate, they control how much of that product is produced. So they get more, less, or none of it. And one way to do that, one place that can happen, is the regulation of transcription. And operons are the classic example. Operons are a common setup, especially in prokaryotes, as a way to cluster genes so that they're regulated together. So more transcription, more RNA, more protein, shut off transcription, and you turn off your protein pipeline. Transcription is controlled by regulatory proteins. You may also know these guys as transcription factors. And there are two types: regulatory proteins that increase transcription from a certain gene, and regulatory proteins that decrease transcription from a particular gene. And that is positive control and negative control. So in positive control, the regulatory protein will bind to the operon somewhere aroundish the promoter, and that is going to lead to an increase in transcription, or transcription turning on. And in some cases, this has to happen for transcription to turn on. And because it's being, kind of helpful in the context of transcription, it gets a helpful positive sounding name: activator. So the activator protein will bind the DNA and help RNA polymerase initiate transcription. Now, in negative control, a different type of regulatory protein will bind to the operon DNA around the operator region, and it's going to block transcription from happening. You'll see a decrease. And so this regulatory protein gets a less helpful sounding name: repressor. Like in so many processes in biology, proteins are doing the work here, but they need to know when. Like, when should an activator bind to the DNA in order to start up or increase transcription, positive control? Or when should a repressor bind to the DNA in order to decrease or shut off transcription, negative control? They need a signal. And that signal is going to be the presence of tiny molecules called effectors. The word effect, it's in the name, right? That tells you these guys are going to cause a change. They're going to cause a change in our transcriptional status. Now, different operons will have different effectors. If the presence of the effector leads to transcription increasing or turning on, that operon is inducible. And because it is helping with increasing transcription, it's helping to get something started, transcription, we call the effector here an inducer. Inducible operons often produce proteins involved in breaking something down, catabolic pathways. They're kept in an off position while the thing they want to break down is unavailable, right? You don't want to make those proteins, they have nothing to do. But when the thing they want to break down does show up, it acts as the effector, the inducer, kicking on transcription of the genes to make the proteins to do that breakdown. But if the presence of the effector leads to transcription decreasing or turning off, that operon is repressible. So we call the effector here a co-repressor, because it participates in that process of transcription going down. Repressible operons often make products that are involved in building a molecule, anabolic pathways. They're going to be kept on in order to build that molecule. But eventually, if you get enough, if it builds up, then that molecule acts as its own co-repressor to go back and slow down or shut off the process of making even more. So when adding effector, if transcription goes from off to on, that's an inducible operon. But if transcription goes from on to off when effector is added, That is a repressible operon. Without adding too much detail here, the regulatory proteins, both the activators and the repressors, have two different forms or conformations, two different shapes. The first is an active conformation. And in that active conformation, your regulatory protein has a DNA binding site that's going to recognize specific locations on the operon, bind to the operon, and influence transcription, either positively, if it's an activator, or negatively, if it's a repressor. The inactive conformation is a different shape, where the DNA binding site isn't available. So it can't bind to the DNA, it's just kind of hanging out. Now, when the effector binds to the regulatory protein, that's going to cause that change in conformation, that change in shape. For some regulatory proteins, binding to the effector activates that protein, but for others, binding to the effector inactivates the protein. And I know that that might seem a little bit unclear, so let's look at some examples. Now that we know what's going on with positive and negative control, and with inducible and repressible, we can sort of mix and match those to get our 4 different categories. I'm going to disappear for a little bit so I have a little more room to draw, because so many exam questions can be made from diagrams of these things, but I will be back at the end. Feel free to sketch along with me. Let's start with the 2 negative systems. Remember, in negative control, our regulatory protein is a repressor. So we'll see that when the repressor is active, it'll bind the operator and block transcription. Transcription's only going to happen when the repressor is not able to bind the DNA. If an operon is inducible, think of its default state as being off, but it can be switched on by adding effector. And in inducible systems, remember, we call that effector inducer. So without inducer, transcription is off. And it's off because the active repressor protein is bound to the DNA, blocking RNA polymerase. But if we add inducer, the inducer binds to the repressor, changing its shape. And with this shape change, it becomes inactive. It can't bind the operator, it's no longer in the way, and RNA polymerase can do its thing. Transcription is induced. The lac operon is the go-to example of a negative inducible operon. Now, in a negative repressible system, our regulatory protein is still a repressor because it's negative. That part doesn't change. But a repressible operon, think about it as being normally on until we add the effector. That means without effector, which we call a co-repressor in a repressible system, without effector, the repressor is inactive. It's not bound to the operator, so RNA polymerase has a nice clear path to work with. But sprinkle in some of that co-repressor, that's going to bind to the repressor protein, changing it to an active shape that's able to bind the operator and block RNA polymerase. So now transcription is off. The tryptophan operon, or trip operon, is a negative repressible operon that you may run into. Positive systems are defined by having those helpful kinds of regulatory protein, activators. Activators can only stimulate transcription when they're active and bound to the DNA. In a positive inducible system, think about the default state as being off. So without effector, without inducer, transcription is off. And if transcription is off, that's because the activator is inactive, not being helpful. But if we add inducer, inducer binds the activator, causing a shape change, allowing it to bind to the promoter and be helpful, bringing in RNA polymerase and inducing transcription. In a positive repressible system, our regulatory protein is still an activator. And in a repressible system, transcription is on without the effector. And that on state means we start with an active activator being nice and helpful, cooperating with RNA polymerase to initiate transcription. But when we add an effector, which is co-repressor in this repressible operon, that co-repressor binds the activator protein, changes its shape so that it can't bind to the promoter. RNA polymerase loses its buddy, and expression, transcription, goes way down. In a big, messy summary here, inducer is going to lead to increased transcription in inducible operons by either activating an activator in positive inducible operons or by inactivating a repressor in negative inducible operons. Both of those cases will lead to increased transcription. And co-repressor will hinder transcription in repressible systems by either inactivating activators in positive repressible operons or by activating repressors in negative repressible operons, causing transcription to fall. If you get overwhelmed, break the categories down. Inducible and repressible are going to tell you what happens when a effector molecule gets added. Is transcription going to go from off to on, inducible, or on to off, repressible? And positive and negative tell you how. What type of protein do you have? Do you have a protein that is helping transcription, positive control, or hindering it, negative control? And of course, drawing it out always helps if you can. Give me a like if you learned something about these different types of gene regulation, and subscribe for even more biology explanations and examples from me, and I will see you next time.